Molecular classification of breast cancer. Our understanding of breast cancer has changed as a result of the discovery of 4 molecular subtypes of breast cancer. This has been made possible by gene expression profiling, whereby the classification is based on the presence of either of the following receptors: estrogen receptors, progesterone receptors, and human epidermal growth receptor 2 proteins. The 4 subtypes of breast cancer are Luminal A, Luminal B, human epidermal growth factor receptor 2, and basal-like. Luminal A, subtype of breast cancer. It expresses hormone receptor for estrogen. Progesterone might be present or not, and it does not express human epidermal growth factor receptor 2. Luminal A characteristic: it has a slower growth rate, it's less aggressive, and typically produces better results than the rest of cancer cells of breast cancer. Luminal B: it expresses hormone receptors for estrogen, human epidermal growth factor, and the progesterone might be present or not. Luminal B breast cancer compared to Luminal A: it's graded higher than Luminal A, it's more aggressive than Luminal A, it has poorer outcome compared to Luminal A. But the best thing, it responds well to HER2 targeted therapy. The third one is what we call the HER2 positive breast cancer. This one, it tests positive for protein called human epidermal growth factor receptor 2, and it does not express hormone receptors for either progesterone or estrogen. What is HER2? Human epidermal growth factor receptor 2 is a protein that promotes the growth of breast cancer cells. HER2 positive breast cancer cells have higher than normal level of HER2. Cancer grows and spread faster than HER2 negative breast cancers, but they are much more likely to respond to treatment with drugs that targets the HER2 protein, like trastuzumab. How do we test for HER2 status? There is one of the tests called immunohistochemistry, that is AHC test, and fluorescence in situ hybridization, that is FISH test. The fourth one is what we call basal-like subtype, also known as triple negative breast cancer. The term triple negative breast cancer refers to cancer cells that lack estrogen hormone receptors, progesterone hormone receptors, and do not produce any or too much of protein HER2 receptors. Basically, all are negative. What are the characteristics? It grows faster than all the breast cancer types, it has fewer treatment options, and it has the worst prognosis compared to other breast cancer types. In summary, this is what we are talking about. If you look at Luminal A, the estrogen receptors are positive, progesterone can be positive or negative, HER2 is negative, and it's known to have a good prognosis compared to the rest. When you go to Luminal B, again estrogen is positive, progesterone can be positive or negative, and HER2 receptor is positive. This is the major difference between Luminal A and B, the presence of HER2 receptors. The third one is what we call the HER2 subtype, and you can see HER2 subtype, the hormone receptors are negative, that is estrogen and progesterone, whereas the HER2 receptor is positive. And the last one, basal-like, triple negative breast cancer, everything is negative. No presence of estrogen, no presence of progesterone, no presence of HER2 receptor, and it has poor prognosis compared to the rest. Those are our references. And thank you, and consider subscribing and share this video.